accomplishments. But for those of you that don't know George, he is currently a professor of genetics at Harvard. He's been pushing technology development on numerous fronts. He was involved in some of the first generation sequencing capabilities and showing proof of concept of Walnut Gilbert. He's also pushed multiplexing, thinking about how to do parallel sequencing and barcoding. More recently, he's developed this colony sequencing technology, which serves as the basis for ADI's Solid platform. He's also been using array-based methods to synthesize oligos, and I think we're going to see a huge growth in the synthetic biology and synthetic genomics area. And it's because of these technologies we can begin to do many things on a cost-effective basis that we couldn't before. In addition to having all these great skills in tech development, he's really been a leader and a visionary in terms of where the field should go. So the Human Genome Project is something that people really question, when should we do it? And by people like George pushing hard, it was able to be launched, and we probably wouldn't be here today without that kind of vision. He has many patents. He's started or had the technology that's built many companies, which is exemplified here on the slide. And he began this program called the Personal Genome Project, and even though we're not health-centric here, this is something that's really pushing, again, the field, thinking about what do you do when you sequence individual genomes, how do you interpret that information, and should we be scared? And I think there's some people that have fear out there because of ethical and legal issues, and he's somebody who I think is trying to show, don't be scared, move forward. So I think we have a great keynote speaker here, and I'd like to welcome them a warm reception, and look forward to hearing your talk here.
It's great to be back at Walnut Creek. Actually, my family had a house here across from the BART station. So I feel that reading and writing genomes is intimately connected, and you'll see slide after slide different ways that they're connected. And I think that I'll try to paint a possible vision of where we can be going forward that leverages where we've been. And I'm indebted, of course, to these governmental and commercial entities that have helped us move things out of the ivory tower and into the real world, and I hope we could continue to do that. So this is over 30 years ago, 1980. This is a purchase order that we placed in Wally Gilbert's lab for some Tenmers, 2 Tenmers. They cost over $6,000 a piece. Probably you wouldn't pay that today.
12 weeks delivery.
Yeah, it was really fast. Yeah. So now on arrays we can build up to easily 244,000 oligonucleotides, which are up to 180 long. And so, for example, $500 will get you 15 million base pairs. So this is 7 logs of progress in 30 years, so it's not too bad. But you'll see that there's even steeper slope coming. So that's writing DNA. This is collaborative research, actually, who later became Genome Therapeutics, which later became Agincourt, that you may know, the newcomers. That's writing genomes. This is reading genomes. Greg Sutcliffe and I, mostly Greg, worked hard on sequencing the first artificial plasmids. So this is kind of a reading and writing story all in itself in 1978. And I estimated around then it was about $10 a base pair. And we were a pretty lean operation, 2 graduate students. And it would have cost more to do it in a more professional way. And now Complete Genomics and others are announcing human diploid genomes sort of at the range of 10 to the minus $6 per base pair. That's a price, not a cost. The cost is actually less than that, and that's already here. This little genome was only 4.6 kilobases and had 5 genes in it. Now this is Moore's Law. I said that we were plugging along with sequencing, synthesis, and telecommunications, all the rest, at sort of a 1.5-fold improvement per year. But then around 2005, we got this change, and this is a logarithmic plot. So this is like exponential on top of an exponential here, where now we're going at about 10-fold per year for the last 4 years, and this will probably keep up for a little while. But gene synthesis, in contrast to oligosynthesis and sequencing, has kind of leveled off, and there's this huge gap of about 4 log gap ready to be filled, just waiting to be filled, and by methods that I'll allude to in just a moment. So you can get these changes from Moore's law, and we should be looking out for them, and when that happens, all sorts of things happen. And so this is kind of like a personal history in here where I'm trying to integrate it with instruments that you're seeing today. These are the Illumina and Applied Biosystems and others are using either polymerase or ligase-based detection of sequence in some sort of either single molecules you'll hear in Steve's talk right after mine, or in most of this is emphasizing a small cluster of molecules which stay in phase for long enough to get some sequence. And we call these polymerase colonies or polonies, and the instrument is therefore a pollinator. And this kind of grew out of the multiplex sequencing that we were doing in the 80s, but Rob Mitra, who's now at WashU, and Jay Shindura, who's now at UWash, contribute to really translating this into sequencing by polymerase and sequencing by ligase with the fluorophores that I don't need to tell this audience about. Now these are 14 of my favorite companies. I try not to play favorites too much, but this is a pretty exhaustive list. And they each have their advantages, and you could imagine a dream technology that combines some of these advantages. So right now, Illumina doesn't have the longest read, but it has a nice compromise of read and cost. Complete Genomics has what we call ROLINEs, they call DNA nanoballs, and these moderate length haplotyping, meaning 100 kilobases where you actually know all the sequences in cis. And they have the lowest price for a genome so far, but that could easily change. The pollinator is open source, and I'll talk about that in a moment. It's the lowest cost device as far as I know. And we have basically large haplotypes, megabase scale, end-to-end on human chromosomes. Enzymatics is not actually an instrument manufacturer, but it actually provides enzymes to a lot of these companies that are about 20 times less expensive with and very high purity. And we can go down the list, but here's a cluster where we have long reads, and I'll give a couple of examples of that in the next couple of slides. So this is something that's been kicking around since, I think, the first DOE meeting, before there was actually a genome project. I was discussing the possibility of doing nanopore sequencing, but I was really delighted to see, this is not our work, but I'm very delighted to see this work from Hagen Bailey's group, published recently in Nature Nanotech, where they can actually discriminate individual bases of not only the 4 bases we all think about a lot, but also methyl Cs. These come into nice little histograms with respect to pore current, and you can see here's the readout real time going along, measuring the current, and showing these 4 different colors here corresponding to the histogram here. And this is the genetically modified hemolysine pore with a beta cyclodextrin buried in it. Now I'm contrasting this and the next slide as showing really out of the box, very different ways of thinking from what we commonly call fluorescent next generation sequencing, which in turn is very radically different from capillary sequencing. So there's another alternative. In addition to the nanopores, here's electron microscopy sequencing. Halcyon is a company that probably most of you haven't heard about, but you'll be hearing much more about them soon. And here you've labeled DNA strands on the order of 15 microns. This can be much longer. A single strand DNA that's quite stretched and labeled with platinum or osmium and so forth. And here's reading out the EM on the order of 0.5 nanometers per base. And William Andrade and others are here at this meeting if you'd like to talk to them. Now, with all this progress, this 4 logs in 4 years and lots of competing companies, we've broken through the monopoly. Why do we need open architecture hardware, software, and wetware in sequencing? And I'll present 2 different ways of looking at this. One is there are certain companies that get the idea of open architecture. IBM PC got it in 1981, and it changed the world, and Google got it in 1998, and this is by no means an exhaustive list. There are many others that get it, and we're working with a lot of them. Rich Terry is our top engineer on this, working with Danaher to bring about this instrument. So one thing is just allowing academics to feel comfortable with an instrument so they can swap out parts and they can write software, not just software that handles the base pairs that come out, but actually that control a machine. Now, the second way of looking at it is that, oh, and this is inexpensive, and there are a lot of beads or reads per run, so that's nice. But the second way of thinking about it is that you can change it. The revolution is not over just because we've got incredibly inexpensive sequencing and it's in good hands in 14 companies. The revolution continues where the sequencer now evolves into a biofab. And by biofab, I don't just mean synthesizing oligos or genes or genomes. I mean analyzing those genomes. And so we can think of is you need certain analytic tools integrated with sequencing, which is now becoming a very common analytic tool, but it's not the only one. So you can think of things like fluorescence activated cell sorters or automated microscopy. So what you can do is the platform that reads sequences you can think of as an automated microscope. That's really what it is. It's reading billions of objects at very high speed on the order of 30 hertz or so at megapixels per 30 times a second. Just by adding a digital micromirror display, or DMD, which we use already for synthesizing oligonucleotides on arrays, you can also use this for release, so selective release of the objects that you're sequencing in the same kind of flow cell. This is a flow cell which can be used for synthesis in organic chemistry, is used for making microarrays to order, but it can also be used, you can also think of it as the same flow cell that we have in a sequencing device. So what if we combine these so that now we can sequence or select, and the limit, that big gap that I was showing in those exponential curves between currently available gene synthesis at about $1 a base and what we should have around 10 to the minus fifth or 10 to the minus fourth is integrating sequencing with the next generation sequencing with synthesis. So in principle, if you have photolabile immobilization of DNA molecules in the flow cell, rolling circle RNAs or DNA nanoballs or beads, or even cells that you analyze with synthetic biology constructs, if they have the properties you want, you can release them into the flow stream and do all kinds of things downstream. So this is an interesting direction they can go. You can have biofab in a box, or you can have it in a big factory, just like you can have sequencing now. You can have a sequencing on your desktop or in a factory. Now, I want to talk about selective genome sequencing. So even as the sequencing costs are plummeting, you still might want to do selective genome sequencing. You go into a metagenome and you might not want to sequence the entire metagenome. You might want to do part of it. Or if you go into the human genome, you might want to just do cancer genes or the protein coding regions or selected regulatory DNAs and RNAs. Now, one way that we've done we've sort of prioritized sequencing is for rearrangements, not just copy number variation, but inversions and translocations, which are more subtle than copy number. Paired ends really needs no introduction to this audience. It's been in pretty much constant use in 2005. Now, for selective, by using synthetic oligonucleotides in red here, selecting out genomic DNA in yellow, there are two major classes. There's hybridization selection, either in solution or on chips, and gap filling and ligation, typically done in solution. And you could do these in very high multiplexing. You'll hear me use the word multiplexing a lot, but you can literally have 258,000 oligonucleotides in one tube. This is something, getting 10-fold multiplexing in PCR is challenging. Getting 258,000-fold multiplexing is so far not anywhere close. But for this method, gap filling ligation, it really works well, and I'll show you some data from that in a moment. Kun Zhang, who's now at UCSD, has done a lot of this in collaboration, and Matt Stilson's lab and others we're very indebted to. Okay, so then how do we do the selective sequencing and how do we get 258,000 oligonucleotides? And every time we change the protocol, we have to make another 258,000 oligonucleotides. This could be an expensive hobby if it weren't for the fact that microarrays can be synthesized by a variety of methods. We typically use the inkjet in collaboration with Agilent. And then we build into it universal primers on the end, which we can then cleave off later by a combination of lambda hexanuclease and uracil glycosidase and DPN2 to get what we really want, which is these two red ends, which are homologous to the genome. And we can barcode, we can bind these to the genome and fill in and ligate, and on the fly, when we amplify these up for putting it on into next generation sequencing, we can add barcodes for the samples. So often when you're doing selective sequencing, the challenge now becomes that you have too few sequences you want. If you can do billions of reads per run, then obviously it's wasteful to fill that up with sequences from one sample, and so you want to do this barcoding. So there are, this is in solution, hybridization selection, and these three refer to the molecular inversion probes. And I don't want to spend a lot of time on this, but you get the idea that we've improved the design. So here going from blue, improving to green, where we're looking at the relationship of the fraction of the bases to the coverage. And so you want this to be as flat as possible. You can see that's improving. Binning is another way of doing it, where you look at which ones are really work very well, do the secondary structure and so forth. Some of that you can predict, some of it you just observe and redo it. Binning helps, and normalization. So these three things, design, binning, and normalization, are making it flat enough so that it really is very similar to doing shotgun sequencing in its efficiency. And so if you can do the genome for $5,000, then you might be able to do the protein coding region for $50. That's not, neither of those are routine numbers, but it's the sort of direction that we're aiming for. Now, in terms of writing and reading, again, are interconnected in that we'd like to harvest the environmental isolates that we've been talking about so much in this meeting, and it's really a tremendous resource. Turn it into functional genomics, and then feed it through some systems I'll talk about for automated genome engineering into things like chemicals, biofuels, drugs, and so forth. And so here's one of the ways that we in our, small way of trying to nudge us along towards functional metagenomics. There's this project called the BioWeather Map, was intended to democratize the collection of samples and engage the public in thinking of this like weather maps, which they already are engaged in. And here is the phylogenetic trees that Jonathan Eisen and others have talked about. And what we did here to turn it into a functional assay is we asked initially what organisms will metabolize lignocellulose, which has toxic properties, toxic to cells and enzymes. And we reasoned that in the soil there'd be a lot of enzymes that might be able to handle these toxins at high concentrations. But we wanted to also test, while we were at it, more well-characterized toxins that might be in lower concentrations in the soil to see if those were there as well. And so we ended up sort of stumbling backwards into this survey of 18 antibiotic classes. These are some of the major classes of antibiotics. And we found that almost every major phyla that we were surveying had representatives here, the phyla are color-coded, and then displayed around the edge the antibiotics that they can handle. And these were not only resistant to antibiotics, but they were actually subsisting on antibiotics as their sole carbon source. And so you could think of this as for us at least, an unexpected catabolic gold mine, and a little bit surprising to people who think about antibiotics even. And if you look at some of these isolates, you would find that there was a big class where they were resistant to almost or all of the antibiotic classes that we looked at. And these were not simply multi-drug pumps, and possibly in part because they were actually subsisting on these antibiotics. So this is work that Gautam Dantas and Morton Summers published in Science. Since then, we've looked at the causative genes underlying this phenomenon of high antibiotic resistance in the environment, and found by sequencing 2 kb clones that basically move some or all of the antibiotic resistance. We've also done much larger clones, but these are much easier to immediately interpret. The larger ones require transposons or something to interpret. Anyway, these 2 kb clones typically have 1 or 2 genes on them, and when we analyze these either from cultured isolates or this dark matter of uncultivated metagenomic libraries, we see a very different pattern. For the cultured ones, they tend to be mostly highly related to known pathogen drug resistance elements, while in the metagenomic libraries, they tend to be, actually there's some points off the chart here where they're not similar enough to even calculate a sequence identity here. So that was an interesting observation, and we're characterizing many of these genes now by functional metagenomics. Now we can use genes from a variety of organisms, move them into E. coli. I mean, I think that the lesson here is that E. coli has the advantage that it has an incredible rate of growth. And so many of the things that you harvest from the wild either have an unknown rate of growth or a very slow one, sometimes days and weeks rather than 20 minutes. And so anything you can move into E. coli has advantages both from a functional genomics standpoint, but also from manufacturing standpoint. And so one of the companies that I founded, LS9, and some others like it, are aiming at making gasoline and diesel for current engines and infrastructure. There's trillions of dollars already invested in this infrastructure, and to think that we're going to overnight switch over to ethanol or hydrogen it isn't necessarily in the cards. And so we want to explore these molecules, some of which are fatty acid derived, or they can be derived from other pathways as well, which are alkanes and alkyl esters. And these have the advantage that maybe even long-term advantage in that they don't require distillation. Instead, there's flotation out of the aqueous phase. They're secreted in the extracellular media. And I include this kind of progress chart not because it necessarily means that much that you make all this, many logs of progress over months, because basically this is just a testimony of how pathetic it was to start out with. But those progress charts are really something that we'd like to get better at, and one of the limitations of metabolic engineering has been the problem of tuning up a large system simultaneously, an entire systems biology problem, and trying to get the design exactly right. So we developed this multiplex automated genome engineering process that I'll describe that allows you to go cycles of genome engineering and selection to either do optimization where you don't necessarily know where you're going, but you know roughly what part of the genomes you'd like to explore, not necessarily all of it, because if you do all of it, you'll start knocking out essential functions. And then sometimes where you know exactly where you're going, and so we have a project where we're making multivirus resistant organisms which might have some utility in biotechnology and agriculture for obvious reasons. So what is multiplex automated genome engineering? Or MAGE. Harris Wang and Farron Isaacs have worked on this, and this is our prototype. This is sort of what the pollinator looked like a few years ago. But this is what this does. Let's see, do I have it here? What this does is in principle, it can use any method of moving DNA into the genome. The one that we found most efficient so far is single-strand oligonucleotide mutagenesis. We've also this has gone from about 10 to the minus 4, which is what we were accustomed to in the past, to greater than 30%, up to 45% if we have another co-selected marker somewhere else. But this can be done without selection at all, and in a sense blind. And what you have is there's 3 tricks. One is that the oligonucleotide is protected on its 5' end with phosphorothiorates, and here we've optimized the number of phosphorothiorates. You can see that that 2 is adequate. We've optimized the oligonucleotide length. It's protected by covering with a phage beta protein. And finally, when it forms a mismatch in the lagging strand where you get, rather than the leading strand in DNA replication, this is the model, when it goes in here and fills that gap that might be left among the Okazaki fragments, the mismatch is formed here, so we have to inhibit the mismatch repair machinery. So phosphorothiorates, optimal length, beta protein, and inhibit mut-S, mismatch repair. And then we have design tools, computer-aided design for the genomics, for synthetic genomes, where you can see this big gap in secondary structure as a function of free energy as a secondary structure of the target, where this is a sweet spot over here where there's very little impact. I'm sorry, on the right, where you get a large number of oligos with very high efficiency. It's fairly flat with oligonucleotide concentration, but we feel that one of the biggest potential bits of progress that we're going to have in the future is increasing the number of DNA molecules we can get into the cell per cycle. Right now it's quite modest, but you'll see what we can do with it anyway. So one of the things we did, remember there's 2 different things we do with it. One is we call accelerated evolution, where we don't know exactly what we want. We want to, say, mutagenize an entire pathway, including side reactions that could take reactions off where you don't want them to go, things that could produce toxic intermediates, all the import and export at each end. And this one here, we're interested in a variety of compounds of agricultural and industrial significance. Here's lycopene. It looks a little bit like the biofuels I showed in the previous one, but it's the red pigment in tomatoes. And we started out with a strain that didn't make any, introduced a plasmid that included this terminal enzyme, and then set upon optimizing it, changing 24 genes, 20 of them up, 4 of them ended up being down, mutations, and changing all the ribosomal binding sites, the RBS, with up to 23,000 oligonucleotides per gene. So there's a very large number of oligonucleotides going into these cells, and in just 3 days of this MAGE, we have a 5-fold improvement to close to the highest levels that have ever been observed in the literature for synthesis of this lycopene. And you can see we went from introducing the initial strain, which produced a little bit of lycopene, was nevertheless very deleterious to the cell's growth. This is downward, meaning that hurting the cell division time. And then after optimization, presumably one of the steps was producing some toxic intermediate, we got a more balanced production line. Even though it was producing more of it, of the final product, it was a better growth of the cell. Here's another example, not from our group, from DuPont, that many of you may know about, kind of a classic example where they spent about 7 years and $400 million of research and development producing this really useful compound, 1,3-propane dial. It's great. They're making it at 90% theoretical yield. This is made for Serona or 3GT, which is used in carpeting and other polymers. This required 27 changes. Remember the last slide was around 24 or so. 6 genes up, 13 down, 8 foreign genes. All of these have to be optimized at their expression levels more or less simultaneously, and this is very challenging. That's why it took a long time. To put that in I use that to put that in perspective. That was 27 changes. Here's a project where we have made 314 changes. So what's the project, and why did we want to make 314 and more changes? Well, we wanted to ask in all these biotechnological projects and health and safety and so forth, what threatens all systems. And these are the viruses that have been increasingly interesting in some of the metagenomics, and certainly is a very significant threat in the day-to-day operations. And to fight these, we could do them one at a time, or we could ask what do they all have in common, or what do they all lack. And what they have is this molecule, which was on my first slide, and one of my favorites. This is transfer RNA that I helped do the crystal structure shown here as a teenager. And this is, and what they lack is the protein synthetic machinery and the genetic code that the tRNA helps do. And so we're making a new translational code with which allows us new amino acids and the safety features I've been alluding to, including multi-virus resistance. This is the genetic code, the one-letter code like tyrosine, and here are stop codons. And next to it is the number of times that it's used in the E. coli genome. Farron Isaac's postdoc has been leading this project. And we started with the rarest but most commonly used amber suppressor codon, or most commonly used suppressor codon, which is the amber, TAG. There are 314 instances of this in 314 genes. So we had to change 314 genes, the end of them from TAG to TAA, and we've now done that. And we're in the process of coalescing these 314 mutations in a hierarchical sense into one genome, at what point we can delete the release factor that normally would be lethal to delete, but now because it's no longer used for the TAG, it can be deleted. And then any virus coming in that uses the TAG multiple times in its genome will be kind of lost and hopefully very sick or dead. And then as soon as we're done testing this, and we'll go on and change other codons. And we can change the code, not just freeing up codons, but swapping codons. And there's a lot of flexibility due to the degeneracy of the genetic code, where 64 codons for 20 amino acids and stop codons. Now that's kind of a transition to protein synthesis. That whole trick is focusing on protein synthesis in vivo and the way that viruses interact with it. And putting in new amino acids is even easier to do in vitro. And you have ribosome display, some of the applications of in vitro translation. Membrane proteins are hard to make in vivo, and they're potentially easier to make in vitro because the ribosomes, et cetera, are resistant to detergents in a way that cells are not. There's a company that does personal cancer vaccines with in vitro protein synthesis. You can label one protein, not the whole cell, for example, stable or radioisotopes or fluorescence and so forth. And there's new chemistry, I'm going to talk about mineral chirality in a moment. And here's an example of ribosome display where you're selecting with a solid phase like a bead for desired protein ligands. And we've worked with Tony Forster on this for a few years. So this is what I mean by mirror chirality, and we think that we're very close now for reasons that you'll see in just a moment. This will give us not just multi-virus resistant, but multi-enzyme resistant, because almost every enzyme, whether it's a lysozyme or a protease or ribonuclease or lipase, has a chiral center that only recognizes one of the two mirror substrates. So here's an enzyme capable of doing PCR, polymerase, and a mirror image DNA in the normal three-dimensional structure. And this is now enabled because of we now can reconstitute a protein synthetic system that works physiologically, as you'll see in a moment. And also other groups have found changes in the 23S ribosomal RNA that permit use of D amino acids, mirror amino acids. And Hirosuga's group has developed a 46 nucleotide ribozyme that will accept any tRNA and any amino acid, allowing you to charge this all 20 amino acids and 19 amino acids in the mirror form. And so here's that same ribozyme again, the ribozyme here and the tRNA here, and they bind by the conserved CCA end to allow us to make any tRNA with any amino acid. And here's the changes that you can make in the ribosomal RNA to accept these D amino acids here at higher efficiency. Now all these things can be improved, but we can see we're going here as long as we can get this system to reconstitute. And these are the 151 genes that we think are required for DNA, RNA, protein synthesis, the central dogma. And in a certain sense that's required for synthetic life, although it's not really our goal. Our goal is high speed and highly accurate in vitro systems, just like E. coli has high speed and high accuracy, around 20-minute doubling time. The real crux of this, we felt, was this molecule here, which has 54 proteins and three RNAs. It's the 70S ribosome. And reconstituting this has been very difficult so far to do under physiological conditions, even though it was done under useful chemical conditions 40 years ago by Traub and Nomura, published the 30S and later the 50S was done. But under conditions that were incompatible with protein synthesis, and we wanted to get this to so that it was, and often the assays would be like polyphenylalanine production from poly-U. So we wanted to get this under physiological conditions, and that's what Mike Jewett did, his postdoc in the lab, showing here production of active luciferase. This is actually a fairly large protein, and here's the reconstituted 50S subunits R50S relative to natural 50S or no added 50S as a control, showing fairly good reconstitution. And here is using synthetic 23S ribosomal RNA together with total proteins which are natural, not synthetic, for the 50S to reconstitute a significantly higher level of protein synthesis than if you don't add the synthetic 23S ribosomal RNA. And again, all of this needs to be optimized, but we've now got our foot in the door, and it becomes an optimization procedure. And I just want to end on one thing that's a little off topic for this meeting, but it's been mentioned a little bit, and I'm here recruiting for the Personal Genome Project. And this but it's very relevant in terms of a way of pushing technology where we're looking at the interaction between genes, environments, and traits. These happen to be humans that rather than trees or microorganisms, but some of the same issues apply. There are some unique issues, which is that there are no open access databases for traits, environments, and genes for humans other than the Personal Genome Project, which is, to me, a little alarming, but at least there's one. And the way we make this open access is we avoid overpromising on de-identification. To put it mildly, we actually promise identification. And in order to do that, they have to get up 100% on an exam. It's not a hard exam. We think that high school education is adequate if you take it enough times. And the idea is that we want to educate people before they consent rather than after they discover that they have a disease. And you'll see an example of that in just a moment. And we hope that this will bring together a large community. It's kind of a crowdsourcing or a Wikipedia type of exercise. It's really not intending for us to centralize this, but to enable it. And it is spreading, as you'll see in a moment. We have low cost and multiple traits. We're trying to get all the kinds of traits you would do for any other organism all in one place, including microbiomes, such as you've seen the functional microbiomics that we did, not only from their environments, but from the individuals themselves and their immune response to them. These cell lines are already available. These are the numbers for the cell lines for each of these individuals. They're already available, as are some of the genome sequence, as much as genome sequences we have, and the traits that we have. And we have approval to expand this to 100,000 volunteers that just really went live a couple of days ago, and we have 10,000 volunteers so far. But we need more. So please go to personalgenomes.org. Now, this is one of the results we already had for one of the PGP subjects, which was we found just looking at the genome a potential case of hypertrophic cardiomyopathy. Here's a major muscle protein in the heart muscle. MYL2 is the gene name, and it has an alanine to threonine at position 13 change. And at the time we did this, we could only find one other case of this in the literature, and it was a case. It was not found in any of the controls that were genomes that have been done. But then asking around, you find that there's a lot of data that's not in any database that clinical geneticists have that they're sitting on, incubating for who knows what reason. But they're happy to give it up if you happen to have another case. And so we find lots of cases of this. And so this individual got an echocardiogram and an EKG and is so far clear of any symptoms, but will have to do this every 2 years for the rest of his life because of the high risk of this. This is a disease that causes athletes to drop dead on the basketball court. Now, just conclusion, any kind of systems biology, not just human system biology, it's very hard to go from the genome to the traits. It's challenging and it's unnecessary since there's lots of things we can do in between. We can measure this bioweather map that I talked about before. Is not just microorganisms, but viruses, allergens, anything that's in your environment. And this is where the DOE has a very interesting complementarity and really overlaps with this. We can go to intermediates like the epigenomics, and we can measure response to the environment. So there's a kind of an environmental component and intermediates that get us from genomes to traits. So each of the Personal Genome Project volunteers, rather than taking a biopsy from every cell in their body, which would not leave them any cells, we take one biopsy from their skin or their hair, which allows us then to form something that's similar to embryonic cells, which then allows us to generate the different tissues by transdifferentiation or reprogramming. And In-Hun Park and George Daly's group and Jay Lee and my group have been automating this. And we use it to look at allele-specific expression. This is incredibly important and a great opportunity for diploid organisms, for those of you who are working on diploid organisms, because you can zero out environmental effects on transcription and the transacting variation that can occur, and you can focus entirely on cis effects, which include copy numbers, which are mostly interesting if they affect transcription, enhancers, promoters, splicing, so forth. And this is by measuring the ratio with any point mutation in cis that's expressed, and it tells you about all the ones nearby that could affect all those cis activities. And here's an example where we've taken this allele-specific expression and we've done clustering of two different PGP volunteers, different biopsies, fibroblasts, bona fide lymphoblastoid cells, and various reprogrammed cells, and you can find very interesting things about variation actually between PGP1 and 9, that, for example, number 1 has a very different response, inflammatory response to a variety of viruses. So just in conclusion, I started out talking about this little synthetic construct that had 5 genes and all the ways that synthesis and analysis fit together, and how we can reprogram E. coli with 4,300 genes directly with this MAGE or multiplex automated gene engineering. And then we can reduce the genome down to 100, we hope, we're on our way to reducing it down to 151 genes, and that's been expedited by our ability to reprogram and reconstitute the ribosome under physiological conditions. And we're moving on to reprogramming Congress here. And 7 billion people, or whatever subset wants to participate in this revolution. So period, full stop. I think there's plenty of time for questions if you have any. Questions? There's a mic back there, yeah.
In the lichen example you gave for the accelerated evolution, you said there were 2 new genes that you didn't...
These were not genes that we discovered. These were genes that were in the literature that we had to introduce to get into the game at all. We're doing this in collaboration with Jay Kiesling's group, and they provided us with those plasmid encoded genes, and then we optimized all the other genes in the pathway, including genes leading away from the desired pathway.
You talk remarkably little about sequencing, and this is why I'm here. I'm curious, as you do all these mutagenesis series or combinatorial type of modifications, you must have to do tons of sequencing.
Right.
Is that all pollinator, and, or do you just do genotyping, or how do you do this quickly?
Okay, behind the scenes, there's a lot of computation and sequencing, and I'm trying to paint a slightly forward-looking picture, but yes, we have to sequence the whole genome, and some of these processes are somewhat mutagenic. So if we disable the mismatch repair machinery, then we build up a slight background of mutations. We typically do as many cycles as we can without looking, because we don't need to. There's no selection. But then we will do routine selections or screens that retains its ability to grow on minimal media so that we don't accumulate auxotrophs. The rate at which we would accumulate auxotrophs otherwise is on the order of 1% for 40 cycles. And then we have genotyping methods that are ligation-based methods, which are lower cost and easier to do than sequencing. And then we do whole genome sequencing to look to see what the whole genome. Now, to some extent, if it has all the properties that we want, being synthetic biologists, we don't necessarily, and it has the 314 mutations that we want or the combinations we have in the case of lycopene, we're happy. But we'd still like to know what the sequence is to see if there are, say, suppressor mutations or potential mutations that could catch us for some other application later on. So yeah, didn't mean to downplay sequencing, but I think the next speaker is going to compensate for that quite nicely.
One final question is, last night we had a keynote on cellulosic biofuels,
Yeah.
and you're really connected to companies and trying to do synthetic biology and produce biofuels. Where do you see this race going? Like, what is the proportion going to be moving forward? Do you see a front runner? I think Chris Summerville was very sobering in terms of how long it might take to get cellulosic biofuels in.
Yeah, I don't want to be unsobering, but I think that we're seeing companies that are already quite good at producing biofuels. It of course depends on the going rate for petroleum, basically. There's some that could be quite competitive if we were at the highest point we were for petroleum already tomorrow, like LS9, that's producing fuels that are compatible with current engines with current feedstocks. I think there's going to be ongoing interest in alternative ways of getting photosynthesis in. So building up complex lignocellulose structure and then tearing it back down again has certain appeal, but it would be nice if you could short circuit that and actually get photosynthesis to go directly to useful biofuels. And I think that's there, if you can solve both the genetic engineering and the bioreactor design, and there are some companies that are, I think, well along on that, then algae and cyanobacteria become quite viable alternatives. So I think I'm a little less sobering than Chris is.
Okay, great. Well, let's thank George again. That was great.